Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 28th January 2021. As we are into the revision mode and we are revising each chapter in the form of test paper 2. Already we have done test paper 1, now we are going for test paper 2, and this paper, test paper 2, in this session we are dealing with chapter number 11, Biotechnology Principles and Process. As I have repeatedly said, biotechnology unit is very important, and there are two chapters. They are not just important for the sake of exams, it is for the general life that we are leading today. Thanks to biotechnology, because of its various inventions and discoveries, our life will be certainly better in future. With that importance, keeping it in our mind, let's go for the question paper. So as usual, first you have MCQs, multiple choice questions. Let's answer first. Question number 1. Lysozyme kills bacteria by destroying cell membrane. ORI, origin of replication, right? Then plasmid, cell wall. I think lysozyme, cell membrane. Then question 2. The group of letters that form same words when read both forward and backward are called. Of course, I gave two examples to you when I was dealing with this chapter through normal videos. One is Gadag. So either you read it this way, G-A-D-A-G, or this way. Second one I gave example, Malayalam. So both backward as well as forward reading spell same. So that should be palindrome. Then question 3. The techniques of using live organisms of enzymes from organism to produce products and processes useful to human. Of course, it's a definition of biotechnology, we know it. And then fourth one, kanamycin does not inhibit the growth of E. coli, amyloliquefacients, and subtilis, and T. aquaticus. Of course, we know it is Escherichia coli, E. coli. It's not bacillus or this. Question 5. To produce copies in billions of a DNA segment, the number of times PCR, polymerase chain reaction, should be done is 5 times, 28 to 32 times, yes, 10 times and 15 times. The answer must be B. Question 6. A recombinant vector with a gene of interest inserted within the gene of alpha galactosidase enzyme is introduced into a bacterium. Why is this method of selection referred to as insertional inactivation? Question 7. Can you recall meiosis and indicate at what stage rDNA is made? Question 8. Which was the first type 2 restriction endonuclease to be discovered? Next, what is TI plasmid? Tumor inducing, right? Question 10. Explain the role of alkaline phosphatase in rDNA technology. What is the source of this enzyme? Question 11. Enumerate the steps that are involved in rDNA technology. Question 12. Can you think and answer how a reporter enzyme can be used to monitor transformation of host cells by foreign DNA in addition to selectable marker? Question 13. Describe briefly the following. Downstream processing. 14. Mrs. Kavita was eager to know the sex of the fetus which her daughter-in-law was carrying. She was so anxious that she could pay any amount for that. The doctor refused to disclose the result of the test. So question 1 in that, what value do you learn from the doctor's act? Question 2. How can one know the sex of the fetus? How is it done? Question 3. Why is disclosing of sex of the fetus banned in our country? Question 15. What essential features must be present in a cloning vector? Now take a paper, start writing the answers, take some say 40-45 minutes and keep the answers with you. Come back to the answers that I am giving. Compare your answers with the answers that I am giving. Question 1, of course, it has to be cell membrane. Either lysosome is capable of breaking the chemical bonds in the outer cell wall of bacteria. Bacterial cell walls contain a layer of peptidoglycan, which is the specific site that lysosome targets. The peptidoglycan layer contains alternating molecules called N-acetyl glucosamine and N-acetyl muramic acid. These molecules form a strong glycan chain that act as the backbone for the cell wall. The link between the N-acetyl glucosamine and N-acetyl muramic acid is cleaved, means broken, by lysosome. Once the chain is broken by lysosome, it results in bacterial death. Question 2. Palindrome. A palindrome is a word, phrase, number or other sequence of characters which reads the same both backward as well as forward. I have given already Gadag and Malayalam, which read same from both front and back. Question 3. Biotechnology, obviously that is the definition. Okay, and then question 4a, E. coli. The genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline and kanamycin are considered useful selectable markers for E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. Kanamycin does not inhibit the growth of E. coli bacteria. This antibiotic is used to treat infection caused by gram-negative bacteria. Question 5, 28 to 32 times. Explanation. To produce billions of copies of a DNA segment, PCR is done, polymerase chain reaction. This is a theoretical consideration as PCR depends on number of factors as optimal priming, salt concentration, enzyme activity, available dNTPC and so on. Ideally, the number of DNA molecules is doubled with every cycle. So the general formula to calculate the number is n into 2 power cycles. N into 2 power cycles. Number of DNA molecules at the end of PCR. N is the number of molecules set in the reaction. And the number of times PCR to get billions copy should be done is about 28 to 32 times. Next, question 6. Due to the insertion of gene of interest within the gene, the functioning of the gene is inactivated. That is, the concerning enzyme is not produced. Question 7, of course, packaging stage of prophase 1 is having that crossing over where genes get exchanged. 8, of course, hind 2 from Haemophilus influenza bacterium. And 9, TI, tumor-inducing plasmid, is a plasmid that often, but not always, is part of the genetic equipment that Agrobacterium tumefaciens and Agrobacterium rhizozenes used to transduce their genetic material to plants. Now see, this is the TA plasmid. Draw the diagram and label each and every part of it. And most important is ORI, organ of replication. And what we are interested is the DNA region, tDNA region. Question 10. The enzyme is used to prevent unwanted self-ligation of vector DNA molecule. Sources from bacteria, BAP, from cough intestine. question 11. Steps involved in rDNA technology. Very important expected question from this chapter. First one, isolation of DNA. Second, fragmentation of DNA by restriction endonucleases. Isolation of gene of interest or desired DNA fragment. Ligation of DNA fragment into vector attachment ligation. And transferring recombinant DNA into host. Culturing host cells at large scale. And extraction of desired product. These are the 7 important steps that you must remember. Question 12. Reporter enzyme can differentiate recombinants from non-recombinants. This is important. Recombinants from non-recombinants on the basis of their ability to produce a specific color in the presence of a chromogenic substrate. An rDNA is inserted within the coding sequence of the enzyme beta galactosidase. This results in the inactivation of the enzyme, which is referred to as insertional inactivation. Insertional inactivation. The presence of chromogenic substrate gives blue colored colonies. If the plasmid in the bacterium does not have an insert, presence of insert results into insertional inactivation of beta galactosidase, and the colonies do not produce any color. These are identified as recombinant colonies. This is how we differentiate recombinants and non-recombinants. Question 13. After completion of biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing as a finished product. The process include separation and purification, which are collectively referred to as downstream processing. The product has to be formulated with suitable preservatives. Such formulation has to undergo thorough clinical trials as in case of drugs. Strict quality control testing for each product is also required. The downstream processing and quality control testing vary from product to product. Question 14. The doctor is devoted to his duties and has professional ethics. And of course, by amniocentesis, we take that amniotic fluid and check for the presence of Y chromosome if the fetus is a male. The process, of course, it is stepwise, and this is how it is done. You can see there is an injection inserted into the belly of the mother, and amniotic fluid is collected, and that amniotic fluid will have the fetal cells, and the fetal cells will be put in that, and look at that. This is what they test. So in the past, there have been numerous cases where the female fetus has been aborted. The moment parents find that along with the 44 autosomes, if they find XX in the fetal cells, they understand that the fetus is going to be a female or girl child. So unfortunately in India, most of the parents were going for the abortion. So thus disturbing the normal cycle and natural ratio between male and female. And also it's a sort of say criminal offense. So that is the reason why this anti-feeling for the girl child is considered as a crime. The number of females to males is thereby reduced, hence it is banned. Question 15. Most commercial cloning vectors have key features that have made their use in molecular biology so widespread. Control of expressions. In the case of expression vectors, the main purpose of these vehicles is the controlled expression of a particular gene inside a convenient host organism like E. coli. Control of expression can be very important. It is usually desirable to insert the target DNA into a site that is under the control of particular promoter. Some commonly used promoters are T7 promoters, lac promoters, and cauliflower mosaic virus with a promoter for plant vectors. Selectable marker. To allow for convenient and favorable insertions, most cloning vectors have had nearly all their restriction sites engineered out of them, and a synthetic multiple cloning site MCS inserted that contains many restriction sites. MCS allow for insertions of DNA into the vector to be targeted and possibly directed in a chosen orientation. A selectable marker, such as an antibiotic resistance, is often carried by a vector to allow the selection of positively transformed cells. All plasmids must carry a functional ORI, origin of replication. So, this is how you need to express your answers, and your answers must be supported or enhanced by suitable, beautiful diagrams. So, keep practicing diagrams and